Thank you. Okay, I hope all of you are back. If you're not, I'm giving it to an empty room, but anyway, so for this next hour, I'm going to talk about the final frontier or space and some of the unique considerations. Thanks, Pedro, for turning on your video. Um, anyway, I'm going to talk about space and some of the special considerations that you need when you're dealing with developing a biomodel in space, because there are some unique things that you need to think about and decide how you want to portray things in the model. So the first thing I already alluded to that I'm going to talk about, and that is how do you put this define the spatial geometry, okay? Define the space that you're going to compute in your model in. And so VCell has a number of ways to help you with that. And I'm just going to use a few examples to show you. So, and then, you can always come back to us for help or use the help menu in VCell to figure out how to do exactly all of these different things. So we provide you a number of tools to add geometry. So if you want to create a new geometry, you can use the same biomodel and use multiple different shapes of the space. But you would replace the geometry, for example, and I'll show you that well, I can show you that now. So you could say you want to put in a new geometry and you can put in 1D, 2D, or 3D analytic equations. You can get it from a file. We can take it from an STL mesh file. We could just paint on a blank canvas, or we could use constructed solid geometry. And so I'll show you examples of all those. And we help you do all of those things. So for example, this case I have here is from our tutorial, one of our tutorials called the FRAP tutorial, in which we've just done a 2D circle, basically. So the expression for a 2D circle here is x times x plus y times y is greater than 100. That gives you a circle of 10 micrometers, okay? So you can, if you say you want analytic and sorry, so if you wanted to add an analytic subdomain, you would add, say, a circle or a rectangle or whatever. You'd put in the center points and the radius, and it would copy this expression, and you could make that your domain. So that's how you can build things out of geometries. You can also build things out of constructed solid geometry. So this is an example where we created a yeast cell with a nucleus by using constructed solid geometry. And you can see each of the domains. This is the yeast cytosol. And this subdomain 1 is defined here. And VCell has some tools to help you create this constructed solid geometry if that's what you prefer. I don't need that anymore. So one of the most common ways to use space is to try and have your model recapitulate an experiment that you've done, say a fluorescence imaging experiment in which you have data that is represented as images. And so what you ultimately want to do is compare the output of the VCell model, right, the output of your simulation with an actual experimental result. So you would like to perform the model in exactly the same geometry as the cell that you did your experiment in. And so we do provide tools, as I showed you earlier, for putting a geometry within VCell. And I'm just going to show you real quickly the beginning of how you do that. So if you wanted to replace this geometry in our multi-app model with a new one, I can make it from file folder. In this case, I have a folder of images that are actually on our website, vcell.org. If you want to practice with this, you can get the same image. I'm only going to take a few because it takes too long otherwise, but let's just take this particular section from this 3D stack here. And it will ask you if you want it at the same size or if you want to scale it a little bit. How many channels do you have? In this case, I only have one fluorescence channel, and I'm only selecting one time point, so I can say okay. And then it'll ask me, what do I want to do with this? And I'll say, well, this is not pre-segmented. This has 255 different gray value levels. So I just want it the way it is. And I'm going to add an empty domain to start with that I'm going to call the cell. And this is a big image. So this is the cell that I want. I can, I've got a lot of tools within VCell now to segment this to tell the software which pixels belong to the cytosol and which pixels belong to the nucleus. So I can smooth it so that it makes it easier to select pixels. I can just select those pixels that are part of the cell and say apply. This is going to be now all those red pixels are where I want the cell to be. And I can add another domain and make it just to pick out the nucleus, for example. I can just show you that real quick. And I can either use the histogram again, or I can even just paint it. So I can just say, well, these ones right here are going to be the nucleus, okay? And so that's now my second domain, right? So anyway, we do provide tools for you to be able to insert your own geometry from images, from experimental images. Okay, so once you have the geometry in there, then you need to think about how you want to position things in space. So if we think about this this PDE model, so let's see where I am. Okay, I'm going to go to a different tutorial to explain this part. And this is a tutorial where we're doing photo bleaching of a membrane species. So I have a species that's fluorescent in the membrane of a cell, and I'm going to localize a laser beam to bleach that fluorescence so that there's a dark spot in the middle of the cell, and then ask how that spot changes because of diffusion of the fluorescent molecule. So in order to model that experiment, this is a classic photo bleaching experiment. In order to model that, I need to assign the laser a position in space. So I have my fluorescent molecule in the membrane, but I'm going to use a species that I'm going to call a laser, just so I can accelerate this bleaching reaction only where I have the laser beam focused, right? So in order to do that, in my initial conditions, I need to place where I want that species laser to exist. And I can position that not only in space, actually, but I can do it in time as well. So this is done using Boolean expressions. So a Boolean expression is one that wherever it's true, it evaluates to a value of 1, and wherever it's false, it evaluates to a value of 0. So in this case, I put the laser beam to be where x is greater than 22 but less than 32 in x, and greater than 35 and less than 35 in y. So I've made a square laser beam in this cell that will bleach, and it'll only be a value of 1 from between 2 and 3 seconds. So I can localize that stimulus in time. So if I go look at the simulation results for this, actually it's best to oh, this is a 2D. Do I really want to do this in 2D? No, this is 3D. Anyway, if I look at this over time, I want to go back to 2 seconds. And we can see that it's a little bit tricky to tell where I bleached, right? I bleached only at the top of the membrane. And I'm trying to get my 3D viewer. There it is. So if I look at the surface here, and I go back to right after the bleach, I don't remember when it was. There it is. Okay, so I bleached the membrane at between 2 and 3 seconds, only at that region of the membrane, and now that fills in. So I can place species in a particular space within the volume, right, within the compartment, the membrane or the volume compartment, by using those Boolean expressions. So does that make sense to anybody? Any questions? Just pipe up if you have some questions, because I'm just trying to hit things pretty quickly so you know they exist and then can ask questions later when you need to use them. Okay, so we can place molecules in space based on Boolean expressions. there's another way where I can place molecules in non-uniform distributions that is can be tied closer to experimental images, and that is using something that we call field data. And again, I just want to show you that this exists and not go into the too complicated a scenario, but this is one model I'm pulling up now that I did with a collaborator, in which they were looking at an actin binding protein that bound to actin. And the problem with this model is that the geometry is pretty complicated. And so if we look at the geometry definition, you'll see the surface view here. This is a dictyostelium, and it's a little complicated. And I had the case where we wanted the actin to be in just a little thin strip around the membrane. So how can I define that as a thin strip around the membrane? Well, there's two ways to do that. Using field data, where I can use an actual image of, say, a fluorescent image of different gray level values to specify the relative distribution of a molecule, or I can use the result of a simulation. So I'll show you both of those just because the result of the simulation also shows a cute little trick in VCell. So if I want to have a species just very close to the membrane, I can actually create a simulation that will create a gradient like Les showed in his very first talk. So I'm going to have a species, a reaction that just creates a molecule from the membrane and then allows it to diffuse in the cytosol or the compartment and then gets degraded. Okay, and when I do that, and if I pick the right situations, then let's go to this one. Oh, I actually have it open already, so let me just pull it up. So what it creates is a reaction, is a species which is very high close to the membrane, which is what you'd expect for cortical actin in these cells, and very low in the center of the cell just by the virtue of picking the right parameters for that reaction. So I run that reaction to steady state and I would get this distribution. So now I would like to use this distribution in order to be the initial condition for another model. This is also very useful if you have trouble getting to steady state. You have to run your model for a while and then want to start new models just once you've already gotten a steady state. You don't want to waste the time finding steady state with having to compute that every time, right? So what I can do is take this distance and actually create a field data. So I can go to the file where it says field data here in the file menu, and I have a manager that manages all the field data that I've created. And this particular field data is this one, 10-fold 5 micrometers. And I can, view that. And what you'll see is that this is, in fact, what I just showed you as the hold on, I just have to make this bigger so I get all the functions here. So this is exactly the result of the simulation that I showed you further, but I've saved this into the field data. And I did that by saying I'm going to create, create field data from a simulation. And when I say create, I can say I want it to be the result of that particular simulation, and then I would say okay. And that creates this field data. Then I can take this field data, which has multiple variables, all the multiple variables, and I can pick one and I can copy that function. And once I copy the function, I can then put it in the initial conditions, the specifications for the cortex, and not the distance one, but the actual model, which is here. And so you have this VC field. That's the function for the field data that I pulled from the simulation. In this case, I multiplied it by a number to make it equivalent to the total actin, so that the total actin concentration in my cell was equivalent to the total actin concentration in my simulation. I can also create field data from an image itself. And in fact, in this particular simulation, that's what I did. So this uses for the field data that I pulled from an actual experimental image. Let's go back here to file, field data. And now I have this particular piece of field data, which is, we can view it, and it's actually a fluorescence image of the field data from a dictyostelium cell. So these are all the Z sections that were associated that I pulled into VCell using our image viewer. And I can use this as now field data so that when I start my simulation here, if I look at the simulation results, which are already open, no, they're not. Oh, yes, they are. Sorry. Oh, no, that's the wrong one. Get rid of that one. I'm just trying to find the results for this. There's some here on this screen, and I'm just blocking them. Well, anyway, this one was actually done. Let me go to this one because I always have to go find myself in Z, right? I have to find the right space in Z, where you can see that now the actin is actually distributed exactly the same way. I can go to the F actin distribution. Is exactly the same way that I specified by that field data, okay? So this looks exactly like the field data. So that's another way of starting a simulation with your molecules not uniformly distributed in space. And in this case, we can align it exactly with the output of an experimental image. Okay. Oh, I'm not doing too badly. So again, if there's any questions, please stop me and, you know, jump in and let me know where you're confused. But so once you've started the simulation and you run it, now you want to analyze the simulation. And again, VCell has a fair number of tools in order to analyze simulations. Oh, I hope I didn't close that. Yeah, so this is again the FRAP tutorial that I talked about before. And we can go look at simulation results from that and see how you might want to. It's okay. It's just sometimes glitchy. Might want to analyze the results for this. So oh wait, this is the wrong one. Yes, there's probably that. I want to use the membrane FRAP one, I think. Yes. Yes, this is the one I want to use. Okay, so here we have our membrane FRAP data again. So now the question becomes, how do I want to analyze this? What do I want to look at in order to understand how my simulation is performing besides looking at this very pretty image? So there's a couple of things that I can do. If I look in the slice view, right, I can find the slice that includes the membrane, which is at the top and at the bottom. So that's kind of hard in XY. So you actually would want to look at XZ. And see, there's where I bleached it, right here and right there. So I could draw a line and create chimegraphs the way Les did, right? So I can plot that as a chimograph. And that will show me how things change along that line over time. So, if I look at one particular spot there, it starts out at this value at 5, and then it's bleached down to a low value, and then over time it comes back, right? So that's one way to do it. I could also average over that line, for example, by doing statistics. So I can take that line membrane, and then it will calculate the mean over that line and the min and the max. So the max, the min, sorry, would show you what's in the very center of that spot, and the max would show you something out on the edge, right? And the mean is averaged over the entire line. But that's actually not really what I want either. When I do this experiment, I actually look at both membranes together. And so I really want to look at the sum of this whole thing projected over space. So we have different tools to do that. So one tool to do that is to use the laser beam. Remember, the laser beam is defined at 1 only where the laser is, right? And I can use that to create something called a the right time, so it's on, yes. So I can create something called a snapshot ROI. So everywhere the laser is a value of 1, it will average over that. It'll include that pixel in an average. So it will include both the top and the bottom in this case, because the laser beam goes all the way through Z. It's identical through Z. So in that case, I can I snapshot the ROI. I can now do statistics, and on this particular set of pixels that I know were the place where it was bleached. And so this is pretty equivalent to what you would do. Oops. So now I'm looking at the laser. Yes, that's when the laser's on, but that's not what I want. Actually, what I want is to look at the fluorescent species here. So now I'll do the statistics of the fluorescent species for that same region of space, and now I get my photobleaching curves. And so this is essentially what you would do, being able to compare the experiment to the actual the simulation result to the actual experimental result. So if I I can look at the numbers for this. So if I just go from the graph to the chart, I get all the numbers, and all these numbers I can then at this point would copy and paste into an Excel spreadsheet in order to do further analysis. However, when you do this in the microscope, it's actually even a little bit more complex, because the microscope changes the value of the fluorescence intensity based on convolution that happens during the optical imaging process. So if you really want to compare this data to an image, you would really like to do it to turn this simulated value into what it would look like in the microscope. And that's called a convolution function. And VCell can actually do that too. So if I go to another model, I will show you that very quickly. Yeah, so I'm back to this dictyostelium model where I was looking at an actin binding protein. And in this case, I've used what's called a protocol. Now, VCell has these protocols that depend on the kind of application you're using. There'll be different protocols for ODEs, for PDEs, et cetera. And for PDEs, the main two are, you can put an electrical phenomenon in, so you can do patch clamps, change the conductance of the membrane or whatever, for electrical events using these protocols, but I can also do something called microscope measurements. So in this case, what I've done is a Z projection, which just sums all of the pixels over Z, right, and puts them into a single value of a two-dimensional image. So it's sort of friend of mine used to call this roadkill, right? This is roadkill of the cell. You've squished the cell all down onto one flat plane, and so summed all of the images. And this is equivalent to what you would see in a wide field microscope. So I can then say I'm going to have one fluorescent probe, and now I can also add up my different fluorescent species. So I have, in this case, I have a binding protein, which is called binding protein that's fluorescent. And I have that binding protein free, but I also have it bound to actin. So those are two different species, but they're this only one fluorescence. When I look in the microscope, all I see, my data is the sum of those two species, basically convolved by the microscope. So I'm going to take these two species and put them, choose these as what's going to be convolved into one species that's called fluor. So after I run the simulation for this, now when I look at the results, which I still can't find, I don't know where I put them. It's not it. I don't know where they are. Sorry about that. So let me go back these back. So if I go now look at my what's called post-processing image data, nothing. This is an activation experiment, so there's nothing at the beginning, and I actually activated fluorescence in only half of the cell, so you can see that now my data is 2D. I only have I don't have Z-stacks anymore. All of the information has been smushed down into one two-dimensional plane. And as it would look like if I was taking a wide field image, and then I can look at this over time. Okay. So, whoops, sorry. So anyway, this basically is a simulated fluorescence image, and it's now directly comparable to what I would see in the microscope. And if you noticed when I was doing this, we can do this not only as a Z projection, but we can also do this as a true convolution. So if you know the point spread function of your microscope, you can do a Gaussian convolution with the same specifications as the point spread function in your microscope. Okay, so that's another way to analyze information over space. So I'm actually going along gangbusters here. Any questions? Again? Just wanted to point out while I have this up, this particular simulation, I actually varied a couple of parameters to run the simulation. So when you create simulations, you can do scan the parameters. And in this case, you'll notice I have four different binding constants, forward reaction rates for the binding. And so I can look at any one of those reactions. Of course, it takes a few minutes to pull it out from the server because these images, all of these data is stored on the server, and I have to pull it over to my home computer. Okay, let's see. So I am actually through most of the stuff that I wanted to do. So again, are there any special questions or anybody want to jump in and ask me questions about space? If not, I'm just going to go through a little bit more about the simulations because I kind of gave that a little bit short shrift this morning. Okay, so let me go back to the tutorial. Which would be here. Okay, so this morning when I finished, we were talking mostly about creating the applications at the beginning, and I had created the 3D PDE and then used a copy of that to create a non-spatial deterministic where everything is mapped to the same compartment. And we just to a single compartment where diffusion is very fast. And I have the different sizes for each of the compartments in there. Okay, so again, the specifications, you just have to start with some initial condition, and then In this case, the simulation ran to, oh, this is mine. Sorry, let me go open a different one. This is the one where I had not run the simulation. So let me open the real one. Okay, so again, here we have the results of this simulation where I can look at multiple species, and we can see that it's come to a steady state distribution somewhere around here, around 10, right? So this is now, I know, the steady state distribution of these particular molecules, and I can actually look at these values and copy these values, and then I could use those values as the initial conditions for a different simulation. So I could make a copy of this one, right? And then I could paste all. So it'll ask me, what do I want? And I can put these in. And so now I'm starting a new simulation, but I would start at steady state. So in that case, this won't go anywhere, right? So, but just to show you that you can, run an ODE to steady state and then use that even as the initial conditions for a PDE if they're uniformly distributed. So that's the ODE, and actually, if you look in the tutorial, we also have an example of a parameter estimation where you're looking to find the forward and reverse rate of one of the reactions based on some experimental data that we can give you. And so you can tell the VCell what data is what, and then you can run this as a parameter estimation. And Pedro is going to talk about that extensively. This is all what is done in Copasi that we've used. And so he's going to go through a lot of parameter estimation, I think, later this afternoon. And so I'm not going to talk about it there, but we do run through that in this tutorial. Also, I already spoke a little bit about protocols. So when you're in ODEs, you can create events with protocols. That is, you can have stimuli at particular periods of time or after something hits a particular concentration, any number of cases where you want something special to happen based on what's going on in the model or based on time. So those you can all do here by creating either at one time, you can do it a number of times. The only thing to remember here is that you need one trigger to turn it on and one trigger to turn it off if you want a pulse. Okay, so if you just put in one event, then, and again, VCell will tell you that, I'm waiting for you to put in the event. You said you wanted one, but you didn't tell me anything. So it always warns you when there's a problem. So that's an ODE. We can then go to the PDE, which I've already shown you quite a bit about. So I'm not going to, again, go into too much more. I do want to point out that there are things called output functions. So if you want to summarize a couple of different molecules, like in that case where I had a fluorescent species that was free and one that was bound, and I wanted to show the combination of both of them, I can create a function that will be simply the expression of both those things. So I can make it, in this case, like say RAN in the cytosol plus RAN C site. So that would be the total amount of RAN in the cytosol. And that would give me a new result in my simulation result. So if I look at the simulation result, there's now a new function here, which I call F, which is actually the combination, the total concentration of RAN in the cytosol, whether it's bound to cargo or not. So you can create all kinds of different functions that help you analyze and look at the results of the model. Okay, so I just wanted to briefly then show you that from that we can also create of the same model a non-spatial stochastic model. So from the ODE, we can make one that is a stochastic model by, again, creating a new application or actually copying it. And again, copying it will keep the relative volume sizes for the different compartments. So that's very useful. And again, this is the same geometry, the same compartment sizes. We can start off. Now here, you'll notice that we have molecules. And the reason we have molecules is because if you're doing it stochastically, concentrations don't exist, right? You're looking at every molecule at a time. So, and in order to do that in VCell right now, you must use mass action kinetics. So that's why this particular model, tutorial model, we always use mass action. So, and it will take you to, for the initial condition, it'll ask you for the number of particles. You can actually look at what, that's exactly the concentration I had used before, right? So now it's number of particles. And when we look at the simulation results, well, so first of all, this is the simulation. And again, you look at the parameters and then there's a solver. And in this case, of course, it's stochastic, so it's chosen the Gibson solver. These other three solvers are hybrid solvers, which will do, which are invoked only when you say that you want some species to be continuous. So if I, for example, once I ran this for a while, had one species that was present in enough molecules that it's better to solve it deterministically within that same equation, I can force it to be a continuous. And in which case this particular one will not be a pure stochastic model. So then when I went to simulate it, I would have to use a different solver, and it would automatically choose one of the hybrid solvers that was most appropriate for your model. It won't do it right now because it actually still has the old math. I would have to regenerate new math in order to have it correctly pick the solver, but I don't want to do that. So I'm gonna see it. It's telling me this is wrong. I can't do that with the mathematics that I currently have. So I'm gonna get rid of that. All right. So then when I look at the results, obviously, of a stochastic model, now you have this sort of stair step, and it looks amazingly like our deterministic model, except that it's a little bit more random, right? It's not nearly so smooth. So, and that's the same thing for a C site. This is RAN site, so you can look at both of them, or you can look at so RAN-C is leaving the nucleus, right, and going into the cytosol. So those are the various transport rates, right, for numbers of molecules now, not concentration. Okay, does that make sense to everybody? And I see there's a little bit of chat going on. Okay, Jim, what is only supported by spatial PE versus particle? I don't know if he's online and can tell me, but anyway.
Yeah, and force continuous with the button that you pushed previously was that would only work for a spatial simulation. Where we have the non-spatial hybrid stochastic is a different type of hybrid. It chooses kind of a normally discrete events are treated as a Poisson process, and then you also have sort of a stochastic process for those for some other portions of the model that are considered stationary.
Okay.
It's kind of there's some kind of hyperparameters in there that choose how the solver makes that choice. But essentially, if there's a large model, it'll run it for a while. It'll notice that some subset of the model is essentially stationary and not changing, and then it can take a large time step until the next rare event happens. It sort of
Oh,
calculates based on this. When does the next rare event happen? Let me just fast forward to that. And when I get to that point, I'll sort of resample that's the stationary noise into some appropriate thing. And then
so
say
do,
now
okay.
it happens.
So would you have to then select that you want the hybrid solver?
If you would like to, you could try it. You could try the hybrid solver, but it will choose all by itself which variables to consider
Okay,
uh differently than others.
okay,
Right?
okay.
Yeah.
Yep.
So the force continuous is only you shouldn't actually be able to push that button, I think.
Oh,
in
okay.
in
I've only
non-spatial.
done hybrids, spatial, so
Yeah, that's where
that's
it's appropriate.
right,
Yeah.
right. Okay, okay. So let's move on to hybrid to spatial stochastics now. And again, this is one where you have to consider space, right? So the geometry is still is our spatial geometry now in 3 dimensions, and that we were using before. So the surface view is here, right? And you can see it has this nice spatial geometry. And then when we invoke again, same initial conditions as we had before. Now, of course, what I didn't mention before with the PDE, but I should have, so I'm glad I reminded to do this. I need to enter the diffusion coefficient, right? Because now we're considering diffusion. So in this case, all of these species are cytosolic species. So the default that VCell picks, which is 10, is appropriate for a typical cytoplasmic protein. But, if you have something that's very big, it would be smaller. Or if you have something really small, like calcium, it might be, 300 or something. So, or IP3, there are certain small molecules that would need a much faster diffusion coefficient. But for most proteins, this is probably about right. So, so you put in the diffusion coefficient, and again, now this is where the force continuous, I can force this to be treated as a hybrid situation if I choose to. And then when I go to the simulations, again, I can start with either 100 particles or 1,000 particles in this case. So if I pick 100 and I look at the results, I now have space. And let's look at RAN-C in the nucleus. So here you can go, there are my particles. So at the beginning time, there should be 100 of them. If I counted up all those dots, there would be 100 molecules. And over time, they're going to come out into the cytosol. And you will see, if I look at RAN-C in the cytosol, now you will start to see some dots in the cytosol. Okay. Obviously, this doesn't provide all that much information. So usually you would go to the statistical data, for example. So this is, again, that curve of the total number of molecules in the cytosol for C. Let's see, here's RAN, the total number of molecules in the nucleus, for example. And again, stochastics are not really all that useful unless you do a number of them, because every time you run the simulation, it will be slightly different. So, so spatial stochastics can take a really long amount of time if you need to run multiple different simulations, including all of space. But depending on how many particles you have, they can become quite a lot. Okay, anything else? Anybody else wants to jump in and ask a question, or? No? Okay, so there's one other thing that I wanted to show you, since I don't think we're ready to go for image J yet. Yeah. Oh, Ezekiel, you are there. Are you ready?
If we want to, we can just go ahead and start presenting.
Well, if you're here, give me, there's one thing that I haven't talked about that I would like to talk about before we jump into the tutorial, but as long as you're here, I'll just call on you in a minute, okay?
Okay, sounds good.
Okay, so the one thing that I haven't pointed out, and I'm hoping Michael will later on, but I also wanted trying to think of ones that a model that would have the right components in it to show you this clearly. Let's go to a curated model. So we have a project that's ongoing, which is trying to help people build model by building little building blocks and enabling you to paste them in. So as Les said, and as other people have said, you rarely start a model purely from scratch. The easiest way is to go find somebody that has already built a model similar to your situation. So the VCell database is very handy. Not only do we have models from VCell, but we also have a lot of models that you can bring in, for example, from biomodels.net, which are all models that are created in a language that can be passed back and forth. And we'll talk a little bit more later in the course about the languages for transferring models from one software to another. These are all so-called SBML models. So there's a ton of models in here that you can just load directly into VCell, and there's also all the models in the VCell database. But we also have created little pieces of models, and so this is one example. If you happen to have a GTPase, small GTPase in your model, you can pull this one in and show it. And we have sort of all the ones that are all the molecules that you need and all the reactions that are based on somebody's data, right? Somebody but what you want to do is you want to annotate this. Where did all this come from? And as you're building the model, you'll need to figure out where did that parameter come from. You'll look it up in a paper and try to figure out, what forward rate constant to use for that reaction. So it's really handy to be able to actually annotate your model with all of these modeling decisions that you made along the way. And so VCell now has a fairly rich system for creating annotations. Now, it's a little extra work to begin with, so I have to admit guiltily that our tutorial models don't have aren't annotated yet, but this just gives you an idea. So in this case, this is a reaction, and we've annotated it with a model, some information from Reactome, for example, or from PubMed describes this, and you can double click that and go to PubMed and find the paper that talks about this particular reaction pathway and where we got those parameters. So you can include all of that in your model for reactions. So, and you can also include text annotations to say this is the reaction that explains the hydrolysis of GTP to become GDP. And in this particular paper, it's said that they got the parameter values from reasonable ranges, whatever that means. We can also click on the species and Michael will explain what molecules are, but we can annotate that with exactly what molecule that is from the CCB database or from Unipart. Because you can imagine if you have a model that's, for example, looking at calcium, you might have, you might call it C, you might spell out calcium, you might call it Ca, you might call it species 23. But so when I'm reading your model, I have no idea what that is. So annotating the model means that I could reuse that model because now I know exactly what species you are talking about when you make that model. So annotations are extremely useful for reusing models. And we're kind of hoping that if we build enough little building blocks that are already annotated, you could string them together and make fully annotated models. That's the ultimate goal. But VCell does have these annotations, and I use them extensively, especially to remember where I got a parameter from. So that parameter came from this paper Y and from figure 6 in that paper. And you will find that it's worth spending the time doing that, because as you build your models, you get more and more complicated, and you start to forget where you got all that data from. It's very important to keep track of where you're getting your data from. And often you can get it from different models, you get a parameter from a different model. Okay, all right, so that will just give us a little more time to talk about the last bit of my spatial sort of modeling in component, which is that, often when you're doing, so Zeke, you're going to jump in a minute. I'll just give a little introduction. So I'm going to introduce my colleague here, Ezekiel Valencia, who's been really doing the development for this part of it. And what we've realized is that comparing simulation output to actual experimental data when you're talking about space is very complicated. So ultimately, what you would like to do is to be able to analyze your fluorescence images right alongside with your output of your simulation, in order to do things like fit parameters. So I can get a diffusion coefficient by fitting photo bleaching data, for example, or how much of my molecules are free to diffuse and how much are not diffusing in this particular case, or what's the binding rate based on a photo bleaching experiment or other kinds of experiments, photoactivation type things. So it's very useful to be able to analyze both your images and the output of VCell in the same way. And I've shown you a couple of ways that we can output data from VCell, but that would, do the simulations in a way that's a little bit more related to the imaging. But ultimately, what we would really like to do is just get the data out into some program that you can, do image processing with the same way you would do for your images. And that was what, that idea was what convinced us to bring Ezekiel on board and have him create a mechanism whereby we can go to our simulation results on the database and simply load them up in image J, just like you would an image, and be able to carry out the same kinds of analyses. So many cell biologists are very conversant with image J. They use it all the time for microscopy images. So we thought it would be really useful to do for outputs of B-cell simulations, and give you a lot more analysis tools. Than there's no point in us redesigning all the analysis tools you could use. So I'm going to turn it over to Ezekiel for a moment, and he's going to give you a little bit of background into this, and then we're going to sort of show you how we can do this. So Ezekiel, if you want to take it over for now.
Yeah, of course. Thank you for the introduction, Anne. That was absolutely amazing, and really emphasized the point that we're trying to take across, which is we no longer want to have VCell be such a closed ecosystem, but allow the power that is VCell's ability to simulate, for example, spatial models, uh, its results to be applicable within other applications. And right now within VCell Alpha, because this is currently still in development, ironing out a few of the bugs and some other quirks that are currently in this process, but within Alpha, there is the capability to export to a specific file format. That file format is called N5, and its focus is on capabilities of allowing exported simulation data to be accessible in other applications, with the twist being that you no longer need to manage the export data yourself. Instead, it's all managed by some URL that you can copy and paste into, for example, ImageJ, and be able to view the results that you've just created. And it's really, there are there's other tools that we're developing as well to help affiliate and alleviate some of the responsibility of managing your simulation data, such as a table to view all the exports and other such information, but those are currently being developed and aren't fully ready to be shown. However, Anne, I believe, currently has a simulation data export within the N5 format that they currently have loaded within ImageJ using an additional plugin that is developed coincided with the support of the N5 file format. And with
Hold
this,
on 1 second, Ezekiel, because I'm trying to, again, I have lost it in my desktop and I don't know where it went.
oh, that's
So
totally fine.
I need to go find it.
But yes, so it happens to, in addition to this newly supported format within VCell Alpha, there is a plugin which allows for, within ImageJ, that allows for it to accept this file format and digest it within ImageJ. You
So,
can, oh, go ahead, Anne.
no, go ahead.
I was going to say, currently, you can either load the whole entire N5 file within Fiji or ImageJ, sorry. Or you can load it as a virtual stack, allowing for larger models to be sort of viewed without completely causing your system to bear the brunt of having to load a large simulation data export. But Anne, can you go ahead and do you have the
Yes, yes, I just picked a different simulation. I still am somehow can't find the, so rather than close and reopen. So this is a simulation of just a three-dimensional cytoplasmic frat model. And if in the, you can view the data, but I'm going to just export the data. And this is an export tab. And we have a variety of different formats to export in. I can do them as different file types. I can do it as ASCII files, CSV files, JPEG, whatever. But this last one is this remote storage compatible with ImageJ that Ezekiel has talked about. And that would be the export. And I just, select it, and then I select what I want to do. I can do multiple channels. So right here, I'm going to do my fluorescent species, which is called APC. And then I can select the time range, and I don't need all that because it's kind of big. And then I just say start export. And so it will create that file format. And there's a number of different compression types, and I'll just do demo 2, say okay. And it will start the process of exporting the simulation data as this N5 image format that ImageJ can read. And also that will be held on, it's now stored not on my computer, but it's actually stored on our servers. So I can copy that location, and then I can go to ImageJ, and there's a plugin for VCell, which we call the VCell Simulation Results Viewer. I'll go to my remote file list and put in that particular location, and then I can open. And actually, this was the one I tested earlier this morning, so they're both here in this list. Now, simulation results change all the time. So if you rerun a simulation, you'll have a new set of results, and these won't exist anymore. So you have to be a little careful to keep track of your own simulation results. But if these are the ones you want, I can just click on it, and I'm going to click on the one I opened this morning, so it'll match with my already baked in one from ImageJ. And this is what Ezekiel was talking about. I can open it either in memory, so put it in memory in my computer, or if it's a really big simulation, it might overpower my computer or take forever to work with. So I can actually open and view the data set just by passing images back and forth with the server, right? So this is all remote. I don't ever have to resave the images again if I want to. Now, this can be a little bit slow, but I picked a particularly small window of time, so I happen to know that this works pretty quickly. So this is my image, and again, this is over time. So there's the Z stack, and there's over time. So this is now an image J. So I'm done with virtual cell. I'm just going to analyze the simulation results in image J. So I can do things like I can adjust the brightness and contrast. And as you'll see over time, let's see, there's where I bleached it. Somewhere in here. It's too far. Let's go back a little bit. Oh, too far. There. Okay, so I can do that on auto. So you'll get a nice bleach region here. This is where I define, remember that the laser should be. In this FRAP experiment, I put the laser right there. So I would bleach the fluorescence only right there. So the fluorescence species is very low there. And then over time, it will fill back in. And I want to measure that because that's my experiment, right? That the length of time at the characteristic time for this to come back in is exactly what I want to compare to my experimental result. So now in this case, I have all these Z stacks. I don't actually want to use all these Z stacks. I want to smush them down into one. Again, I want to do a Z projection. So image J has lots of capabilities to do all this stuff. So for example, I can take my stacks and I can do a Z projection. And this is where Ezekiel's freaking out because I promised him I would do this in memory and I'm not, but I'm pretty sure that it's going to work. So we're going to do it. And it's going to flatten all those just like I did in VCell. And now see, I have this lovely image that is the sum of all those in Z. Now it doesn't actually display it very well, but oh, that's interesting. Oh, I guess that's right. So you will notice when I look at the data points here, right, that I'm not looking at a grayscale image per se of 0 to 255 for an 8-bit image, but I'm actually looking at the actual concentration values that were determined by VCell. In this case, this is the initial one for this Z stack, right? And this is the value when I've summed over Z, but it's the actual value. It's not converted into an image value. It's the actual value is retained in the image. So this N5 format also has the ability to maintain the floating point values in the image. So now what I really wanted to do was I wanted to measure what's going on in that region. So, I have all of image J here to work with. So I can, create regions of interest. And I can then measure that for the entire time series of the stack by, again, just using image J functions. And so, again, you can just measure the stack and I can go and that's a mean value. So then I can plot that. And I can plot the slice versus the mean value and get, this is a standard classic photo bleaching graph, right? Where it starts out high, I bleach it, and then the fluorescence comes back because the molecules are diffusing around and the bleached molecules move out and the unbleached molecules move back in. And so the little spot goes away. So all of this I can do in image J now. And if I had my real cell here as well, or I had 15 different simulations, with 15 different diffusion coefficients, I could generate 15 different graphs and see which ones fits my data the best, for example. So anyway, this is going to be, I think, a really important step forward for VCell, being able to create all these results. And the other thing that's really handy dandy that I should point out is that you can ImageJ also has an enormous amount of scripting options. So, which I can't quite remember where they are. Where are they, Ezekiel? How do I get to the scripting?
Go to usually plugins. Scroll up.
Oh, I do that all the time, sorry.
and then you can either write macros or um, run your own um,
Right.
separate file outside of ImageJ.
Right, yeah. So you can create macros and run them, and I don't have any in here yet. So, but anyway, you can. There's a variety of scripting languages that ImageJ can do, and I'll let Ezekiel
Yeah,
reach up with a
I
little
c-
bit more about sort of where we're going with this in the long run. So again, just to stress, we don't have this in release yet. We hope it will be quite soon, but so this is up and coming, and we'll let Ezekiel take it further.
yeah. So thank you, Anne, for that wonderful demonstration of what can be done currently with the N5 file format and the ImageJ plugin that sort of follows with it. And so within ImageJ, there's a little bit more technical talk. There's a sort of scripting language that you can utilize within it, and it allows for you to manipulate the image data that's currently being displayed on Ann's screen. Um, and with that, because of the way the plugin is written, it's using sort of uh, a universal standard for how the images should be presented and loaded. Now, because of that, you can access the images whichever way you want. And in addition to that, instead of having to write your own scripts, you can try and utilize other plugins within the ImageJ ecosystem to be able to sort of analyze the images that you've loaded in and sort of manipulate them however you want. In our demo, we're planning to automate the whole entire process, but there is a little bit of a learning curve towards the ImageJ sort of scripting language, since it has a sort of semi-custom language to ImageJ itself that allows for macros to be executed along with um, along with other sort of functionality hidden within it. And one of the future capabilities is, as of right now, you can see there's an export table available within the ImageJ plugin. However, still not fully um, it's still not fully capable, at least within other platforms. I know that within Linux, it's fully usable. However, it seems like varying with variation between operating systems, it's not all the way there. But it's essentially used to help you keep track of all of your exports that you've done. Um, because using the URL is nice if you want to share it with someone, since these URLs are not um, individual sort of, it's not tied to any individual. It can be shared as long as you have the link. Um, but that's also somewhat of a nuisance because if you have a lot of exports that you've recently done, it's not necessarily very easy to copy all those links, open them all up, and keep track of what you've opened. And that's where the export table comes in, both within ImageJ and an additional export table within the VCell application. They're both sort of tools that are currently being developed in addition to all of this to help alleviate some of the burden that's affiliated with dealing with, for example, another file format, HDF5 or any other that you can export within VCell. Um, and then in addition to all of this, some of the more future and long-term prospects would be um, opening up this file format into different applications. Now, with the infrastructure that's currently in place for the N5 file format, it is not only coupled to ImageJ, it's just the first sort of example of what we're implementing. Instead, if anyone in the future decides to, either VCell itself or a third party, support the N5 file format for their application, because of the way we set up the way the format is served, it can be accessed by anyone that supports the standard of N5 and S3 protocol. Now, in more of a down to earth application example, that would be if you want to do any analysis within Python. It could be potentially capable of supporting the N5 file format. Actually, let me restate that. It is technically capable of supporting the N5 file format. It's just not supported or encouraged by VCell currently. And the same can be talked about within other sort of potentially useful locations where a URL would be more of a convenience than having to load an entire exported file. So, for example, Jupyter Notebook, instead of having to try and insert a HDF5 file with some additional libraries and some additional support, you can just take the URL that is affiliated with your N5 export, and you can then load the sort of simulation data within a Jupyter Notebook. Now, depending on how you want to use that data within the Jupyter Notebook itself is up to you. Either use it to perform some sort of statistical analysis throughout the notebook and slowly yet surely sort of create plots and graphs, or simply create a visual representation of the data that's available within the Jupyter Notebook. All of that is potentially possible, but also not currently implemented because our focus right now is on ImageJ. But the concept of this is to help and sort of branch out what is the powerful tool that is VCell and its capability for simulating different interactions. And to be more specific towards the N5 file format and how you export it, within VCell, each simulation is correspondent to one N5 file format. And, or sorry, not N5 file format, one N5 file. And with this, it allows for a easy tie between what is exported and the actual and the actual simulation that created all that export data. But hopefully in the future, we can deliver a higher quality product, because as of right now, some of the visual representations of the plugin and other parts of it may change in the future since it's still in alpha. But thank you for your time, and I believe I'm about to run out of time. So my final statement is that within VCell, again, we're trying to sort of branch off from keeping all of the simulated data within VCell itself, allowing for the end user to decide how they want to interpret that data and what tools they want to do it with. So that's the N5 file format and its application within ImageJ. Thank
Right,
you.
thanks, Ezekiel, that was fabulous. So, again, this is just, you know, some of the difficulties in working with space. We're not just dealing with values. over time, we're dealing with X, Y, Z dimensions as well as time. So it becomes much more complicated to do these analyses. And this is one way that VCell is hopefully going to make it much easier in the future to do that. Okay, so I'm done early, which is, well, no, right on time. I was gonna say, I don't think I've ever been done early. But anyway, if there's any questions, please feel free to ask. And also for those of you with projects, if you have spatial projects, obviously we can talk about that more in the the groups when we get together in the breakout rooms. So I'm gonna stop there